Welcome to the endoplasmic reticulum. You have learned about many organelles. The endoplasmic reticulum is unique because it is the cell's manufacturing and distribution center, with two completely different specializations in the same structure. Here is an amazing fact. Your cells contain a maze of interconnected membrane tunnels called the endoplasmic reticulum. If you stretched the endoplasmic reticulum from a single cell, it would extend over 1 kilometer in length. This network has two distinct regions with completely different jobs. One region is studded with tiny factories called ribosomes and looks granular under the electron microscope. The other region is smooth and empty. Same organelle, two different jobs. Here is an amazing fact. Your cells contain a maze of interconnected membrane tunnels called the endoplasmic reticulum. If you stretched the endoplasmic reticulum from a single cell, it would extend over 1 kilometer in length. This network has two distinct regions with completely different jobs. One region is studded with tiny factories called ribosomes and looks granular under the electron microscope. The other region is smooth and empty. Same organelle, two different jobs. The rough endoplasmic reticulum, or RER, has ribosomes attached to its outer surface. These ribosomes synthesize proteins from messenger RNA instructions. The proteins are made specifically for export from the cell, like enzymes, hormones, and antibodies, or for insertion into the cell membrane. The rough appearance comes from the bumpy texture created by all the ribosomes. The smooth endoplasmic reticulum, or SER, has no ribosomes. It specializes in synthesizing lipids and steroids, and in detoxifying drugs and metabolic waste products. In liver cells, the SER is especially abundant because the liver must break down toxic compounds from the bloodstream. Different cell types have different ratios of RER to SER depending on their specialization. Let me trace the journey of a protein through rough ER. Step 1. A ribosome synthesizes a protein from the genetic instructions in mRNA. Step 2. As the protein is being made, it is threaded into the RER lumen through a channel called a translocan. Step 3. Inside the RER, the protein is folded into its correct three-dimensional shape. Step 4. Sugars are added to the protein in a process called glycosylation, which happens only in the ER. Step 5. The finished protein is packaged into a vesicle. Step 6. The vesicle buds off from the RER and travels to the Golgi apparatus for further modification. Here are the key facts distinguishing RER and SER. RER equals rough ER, which equals has ribosomes, which equals makes proteins for secretion. SER equals smooth ER, which equals no ribosomes, which equals makes steroids and detoxifies drugs. Both are connected to the nuclear envelope, forming a continuous membrane system throughout the cell. RER is more abundant in cells that secrete large amounts of protein, like pancreatic cells and plasma cells that make antibodies. Your NET exam will test whether you can distinguish between RER and SER and explain what each one does. The key insight is that the presence or absence of ribosomes is the structural difference. A quick memory trick is rough equals ribosomes equals proteins, smooth equals steroids plus detox. The most common mistake is thinking that RER makes all proteins. It does not. Cytoplasmic ribosomes not attached to ER make proteins that stay inside the cell and do not need processing. Let me summarize the 5 key takeaways. First, rough ER has ribosomes on its surface and synthesizes proteins for secretion and membrane insertion. Second, smooth ER lacks ribosomes and synthesizes lipids, steroids, and detoxifies drugs. Third, smooth ER is particularly abundant in liver cells. Fourth, both RER and SER connect to the nuclear envelope to form a continuous membrane system. And fifth, proteins made on RER are processed and shipped to the Golgi apparatus via transport vesicles.